Good evening, everyone. Thank you for coming to the Connecticut Valley Section American Chemical Society webinar. This evening, we are honored to have Dr. Christian Brückner talk with us about his talk on eggshells. As the American Chemical Society, we're very happy to, welcome everyone. If anyone has any questions during the chat, during the meeting, we ask that you post them on the chat and we will be monitoring them as we go throughout. Now, I would like to introduce Mark Peasy, who will be introducing Dr. Brückner.
Hello, everyone. Welcome. Good night. Good evening. It's my pleasure to welcome tonight's speaker, my colleague at UConn, Professor Christian Brückner. I don't want to take too long with this overture because I want to leave the time that he needs to tell his egg-citing story. Besides, if I gave him a proper introduction describing his contributions to the synthesis, photophysical characterization, and novel applications of porphyrin analogs investigated in his lab, you'd say, oof, I leave some chemistry for the rest of us. Instead, I simply say that he is an engaging presenter and we're in for a real treat. With that, I say, a huevo we go with the chemistry of eggshells. Christian.
Thank you. And I will immediately share my screen here. And then we'll see whether it worked. So do you see just one screen? Somebody say something?
Yes, okay. We see your screen. Thank you.
All right, good. Thanks, you both for the introduction. And I have tried to get all the egg-citing and all the egg-related words out of the talk. But Mark did it wonderful and brought it in all different languages, so I don't need to continue this. Thank you really very much for the invitation and the hosts of the meeting for setting all this up. And I'm kind of nervous about this talk because usually I talk about really my expertise, and I'm not an eggshell expert. I'm not an egg biologist. I'm not even a biologist. I'm a failed biologist, if one wanted to take it really seriously. I started studying a few semesters of biology, but then went back to chemistry and never looked back. But I've always been really a lifelong fan of all things birds. I have picked up every hatched eggshell that I could find in the garden, every feather, every bone, everything that I could find. But I've never really dealt with it in a serious way until about 10 years ago, when my specialty area in chemistry intersected much more in a much more scientifically serious way with the chemistry of the eggshell pigments. I'll tell you a little bit about it. But instead of giving you a completely detailed talk, I've compiled a whole range of beautiful eggshell images, mixed it in with some trivia, some old contemporary science, some deep science images, and then amalgamated this into a presentation for the scientifically curious. And I hope you will enjoy the imagery. Don't pay too much attention to the details. I give you really a broad overview. We'll talk about the details later on. And so I call this the Egg-citing chemistry, the chemistry of eggshells and eggshell pigments, and a little bit of biology. So I am an expert in the chemistry of porphyrins, and the porphyrins are these tetraperolic macrocycles that are also often referred to as the pigments of life. And my real science deals with taking one of these rings and breaking it open chemically and then stitching them back together, and in the process replacing a carbon by an oxygen and nitrogen, or leaving the carbons out altogether. And this is an entirely different way how to degrade a porphyrin than nature does it, as you will see in a little later. But porphyrins are a part of the pigments of life because we need them to live. Hemoglobin, we all know, is red. Our blood is red because of hemoglobin. And hemoglobin is a big protein, and there's a prosthetic group in the center of this protein, and that is a porphyrin. And it is this porphyrin iron complex. We all know iron is in blood. And so this is the porphyrin ring, and there is some decoration of the porphyrin around it. And this particular porphyrin we call protoporphyrin 9. When it's coordinated to iron, we call this the heme, or the protoporphyrinato iron 3 or iron 2 complex. That is really what makes tissue look red, and blood really deep red. And so what we learn from this, porphyrins are really strongly absorbing molecules. That makes them so appealing to me and many others. But their color is rarely being utilized in nature as a color. I think the red color of blood is just ancillary to the function of blood, but the red itself doesn't necessarily have a function. Eggshells, we all know eggs, right? We know there are white eggs, we know that there are brown eggs, and depending on where you live, there is a preference for the eggs, right? If you live in the Midwest, it's very hard to find brown eggs. If you're living in California and in New England, you have really easy access to brown eggs. And that's really just a regional preference. There is a very, very, very large number of eggs being produced a year in the United States. I just looked this number, and I had to copy it in just 15 minutes ago because it says there are in excess of 110 billion eggs produced per year in the United States. Most of them are white because the white leghorn is the most efficient breed of converting feed to eggs. And if you're producing 100 billion of anything, just a very tiny advantage of converting feed to product makes a difference. And so we're willing to, for those areas that prefer brown eggshells or brown eggs, they're willing to pay a few cents more for the brown eggs than the white eggs. Often they're also associated with a little bit more natural or so, and it's not necessarily true. Nutrition-wise, they are the same, but one is white and one is brown. And the brown eggs are brown because there is a dye in it. And the dye is this protoporphyrin 9. It is this pigment that we have in our blood, except there is no iron in the center. It's the free base protoporphyrin 9. And this is where my expertise in porphyrin chemistry and reading porphyrin literature and eggshells intersected. It's nothing new. This is actually very old. 110 or 115 years ago were the first reports of a porphyrin found in brown eggshells. They were extracted by a researcher in Germany, Hans Fischer, who then went on to win Nobel Prize in 1930 for his study on the chemistry of the porphyrins, the pigments of life. And so initially he isolated a pigment, a porphyrin, from eggshells, and he called this the OO porphyrin, the egg porphyrin. And later on, he realized that when he introduces iron into the OO porphyrin, that he then obtained hemin, and he was able to extract the hemin from blood. And so he was the first to really show this connection that whatever is in the eggshell is really the protoporphyrin 9. That's the basis of heme. And so I have read about this not long after I got introduced to porphyrins, and I took that in and I remembered this, and I was surprised at first that when I mentioned this at porphyrin conferences, or I don't know when these things come up, and that hardly anybody knew about it. And I thought, oh, this is weird. This is well known. I should write about this. And we started working on a Journal of Chemical Education paper where we extracted the porphyrins from eggshells, and we did some interesting chemistry, some photophysics, some absorption, emission, and chemiluminescence assays with that porphyrin extracted from eggshells. So that was our first foray into eggshell chemistry. So, but if you go in the store and you look, for example, here at quail eggs, they also contain protoporphyrin 9 in just a splotchy form, in just a different shape, a different distribution, but it's the same pigment, and it was also very well known. The same with seagull eggshells. In fact, the first porphyrins isolated from eggshells came from seagull eggshells. There was a big seagull eggshell festival held at the Baltic Sea by Hans Fischer, and everybody got free seagull eggs to eat. You just had to deliver the eggshells that he then took to Munich and extracted, and that's how the whole research started. But one thing is very surprising about the fact that there is a porphyrin in these eggshells, and that is, it's very rare that porphyrins are being used as a colorant, as a dye in nature. And it is probably because free-base porphyrins are fairly phototoxic. And that's also something that is well known. Meyer-Betz, 1912, he injected himself with a close derivative of this protoporphyrin called hematoporphyrin, and he just sat down and waited for any effect to happen. Nothing happened, nothing happened to him. So then he concluded, oh, that's a safe thing to inject. And he went out, exposed himself to the sun, and then immediately experienced a violent phototoxic reaction. He swelled up. He almost did not survive this. And the swelling and the photosensitivity that he experienced was still, he was still affected from this months later. And so this is one of the things that people are interested in contemporary porphyrin chemistry, and that is using this to affect tissue that you do not want. For example, in a cancer therapy, you could think of injecting a porphyrin, shining light on it, causing this photosensitization reaction to the extreme to kill the cell and therefore treat the cancer. And this is being used, it's being used in the clinic, and that is what drives a lot of the interest in contemporary porphyrin chemistry. And this is also why I thought, oh, that is interesting. Porphyrins are in eggshells. Is there any phototoxicity effect in these eggshells that is beneficial to the birds? And so 10, 12 years ago, I decided to team up with somebody in this animal sciences to investigate this. And we realized very quickly it's very hard to grow any microbes on eggshells. And there were a number of reasons we abandoned the project. Very shortly thereafter, there was a paper that came out that said something about the photodynamic antimicrobial activity of avian eggshell pigments. But it doesn't say anything about whether it also works really on the eggshell. It's just the pigments extracted from the eggshells. Anyways, I thought it was still interesting to look into it. The photosensitization process is working as follows. You have a ground state porphyrin, it absorbs light, it gets into an excited state, it can give off that light in the form of fluorescence, or it can go a different path, end up in an excited so-called triplet state, in which it can react with the triplet oxygen that occurs naturally in the atmosphere. They both exchange their spin and energy, and this generates singlet oxygen, and that singlet oxygen is as toxic as its neighbors in the periodic table. Chlorine, fluorine, super reactive, and that is then why photosensitizers are toxic. We also know that there are some antibiotics, for example, tetracyclines, that when we take them, we are advised to stay out of the light. And that is exactly the same process. These tetracyclines are photosensitizers. So the question is still, is it really, does it have any phototoxic effects? By now, I'm much less convinced that there is, because when you pack up porphyrins in a dense packing, they don't generate singlet oxygen anymore. They have other ways to relax from an excited state. But to me, it was still intriguing, and this was the start how I looked into this. Are there other porphyrins that are being used as pigments? Yes, there are, but they're really, really rare. Here is a free-base porphyrin, uroporphyrins 1 and 3. These are precursors to protoporphyrin in the biosynthetic pathways towards protoporphyrin 9 that are found in some shells here of some seashells. But it's really rare. And the beauty is this is on the left side, you see the color of the seashells, and on the right side, you see them lit up with black light. And so those that give this beautiful red fluorescence, they contain the porphyrins, and this just reflecting the black light or the blue light, they don't contain any porphyrins. So this is one pigment, the porphyrins, in the eggshells. But there is another pigment in the eggshells. And it is this pigment that makes this absolutely beautiful robin's egg blue, this Tiffany box blue. And these are so-called biliverdins. You have seen probably the eggshells, you have seen them breeding on the windowsill, or you have seen the discarded eggshells. A lot of songbirds, starlings, thrushes have this beautiful, beautiful colored eggshells. And the biliverdin and the heme are very closely related, and they are closely related in the following way. Here's the heme again. There is an enzyme in the body that is called the Heme Oxygenase that can cleave the heme at this apical position here, takes the iron out in the process, establishes oxygens in the place where there was a single carbon, and generates this open ring tetraprolic pigment. That is the biliverdin, and it's called the biliverdin 9 alpha, indicating where it was cleaved, and the 9 that it was derived from the protoporphyrin 9. So now we have 2 eggshell pigments, and the amazing thing is that there are some eggs out there that really have this biliverdin encrusted in a thick layer on them. One of them are the emu eggshells. And so here's a photo. This is my hand. I'm holding an emu eggshell. We order now these eggshells from an emu farm in New Mexico. We get 5 kilograms of eggshells, of spent eggshells at a time. We extract them. We get a lot of biliverdin from this, and then we do some synthetic chemistry with biliverdin. Very convenient way of isolating this. We also wrote another Journal of Chemical Education paper on this, just the isolation of biliverdin 9 alpha, and its characterization by NMR and UV-Vis, and do some experiments. A very satisfying chemistry to do, very easy, very straightforward natural product isolation. Usually biliverdins are occurring in the liver, and so on, very hard to isolate from these biological tissues. So here we are. The white shell is just the calcium carbonate shell. We have the protoporphyrin 9, the brown shells, and we have the blue shells, the blue-green shells with the biliverdin. It was all very well known. There's a beautiful review that's already 50 years old, and there was nothing new about this. You can also go now to the store, and if you go to Whole Foods or so, you see the Easter eggers. You see some eggshells that are a little bit more greenish and brown and white, and a little bit more colored than the just brown and white eggs that you usually see, and they contain the biliverdin. And in fact, if you take a little bit of biliverdin and a little bit more of protoporphyrin, you can start mixing them, and you get different colors. And that's easy to understand, easy to see. Just somebody, one of the audience members sent this to Rebecca, this picture. I found it then on Instagram. I thought it was an absolutely wonderful image. And it is an image of all the various eggshell colors produced by hens, by chickens. And so we see only white and a little bit green, a little bit brown, but they are really available from really dark green and really blue-green and various colors in between. And that's also long known. There is a very large color variation. This is probably an old poster. I don't know if it is a really old poster or a poster made to look old, of the various breeds and the various colors they do, and which eggs get produced by which hens. Well, there's an easy way to tell. You just have to look at the earlobe of the hen that lays the eggs. If the earlobe is brown or dark, it will lay brown eggs, and if the earlobes are white, it's going to be laying white eggs. And so there are white feathered hens out there that have brown earlobes. So white feathers are not the determinant for the eggshell color they are laying. So there is a whole variety in the hen's eggshells, but there are way more bird species out there, and there is a stunning variety of eggshell colors, patterns, sizes, and shapes out there. And that I always found fascinating. And how can you not be fascinated when you look here at this image, for example? We'll go into color, pattern, size, and shape one by one. Color. So we have two dyes. We can mix these two dyes and we can generate a whole bunch of colors. But how many colors can I really generate with two dyes? Well, it turns out not that many. There's a paper in which the perceptual color space is mapped out here, and on the dots are the known eggshell colors. And you see only a very small space of the colors that a bird could see are occupied by the eggshells. Whereas when you look here at the avian feather colors, it occupies a much, much, much broader space of that color space. And so the whole paper was called Not So Colorful After All. The paper was written by Daniel Hanley and Mark Hauber. And Mark Hauber from the University of Illinois, used to be at SUNY Hunter College. He is one of the experts in eggshell coloring and everything having to do with colors and birds. And so is Dan Hanley, now at George Mason University. So if you really wanted to get expert papers on the subject, these two people, look at their papers. Always beautifully illustrated, wonderful papers. So in this Not So Colorful After All paper, there's another way to look at the color space, and you look at which portion of that color space is occupied by the eggshells, and you also see that there are some outliers. And I was really interested in these outliers, and so here are some of the outliers. These are the so-called Tinamou eggs. The Tinamous are South American birds. I'll show you in a minute a picture of them that lay these absolutely stunning eggs. They are as shiny as they are presented here. I'll get into the shininess in a second. They are not polished, and they have these really non-conspicuous colors. So here is a crested Tinamou. It's a very non-assuming bird, smaller than a chicken. Live in a broad swath of South America, in forested areas, and they lay these eggs. And so in conversations with Richard Prum from Yale University, a real expert in the biology of color, not just the color of eggshells, he mentioned those Tinamou eggshells, and a little earlier, Dan Hanley had contacted me to get some of our eggshell pigment extracts, and we all stuck our heads together, and then we thought, let's look whether we can discover anything about these Tinamou eggshells. And so we procured some, we extracted them, we had these green ones, we had the chocolate colored ones, and indeed we found two new eggshell pigments: uroethrin, a tripyrrolic pigment, and bilirubin. And if you look at the color of one and the biliverdin, the second pigment in there, you mix them, you can, and you look at the ratio of what we find by analytical means, the ratio of these two pigments, then you get to the solid line, and then you measure really the color of the eggshells. You get to the dotted lines, and you see, oh, there's a good match. That was the discovery of two more eggshell pigments, and it really expanded the color gamut of eggshell pigments. And it makes sense that there are these two other pigments in there. You have the heme, you have the biliverdin conversion, and if you go now from biliverdin to bilirubin, it's just a reduction of the system. Sorry, the double bond here is gone. More hydrogens are here in the center. And here's from this JChem Ed paper, here's the color of the biliverdin, and you reduce it, and then it turns to this yellowish bilirubin. And so that makes sense that it's bilirubin in there. This whole heme to biliverdin to bilirubin conversion we are very well familiar with, and that is when you get a bruise. When you get bruised, you have some heme penetrating your tissue. It's red at first, and then it starts turning greenish as the heme gets broken down to biliverdin, and then it turns yellowish before it dissipates, and that is the conversion from biliverdin to bilirubin, and then the bilirubin gets solubilized and then flushed out of the tissue. So you have seen this chemistry on your own body. Where is the uroethrin coming from? Well, it's a triperolic pigment shown here. And this is just a further degradation of the biliverdin. You take the biliverdin, you just cut off one of those perolic units, and then you end up with this uroerythrin. You cut off yet another perol, and then you end up with these diperinones that we also found in the eggshells, but they are very little color. They do not really contribute to the color, but they show that this degradation process is being utilized for the production of the uroerythrin. And so that was all part of our publication from last year. The shininess, why are these eggshells so unbelievably shiny? And it turns out they're so shiny not because they are oiled, but because they are nanostructured surface. If you look at the AFM image of a regular eggshell, here the Gallus eggshell, then you see a certain roughness. But if you look at the Tinamou eggshells, they are significantly smoother, and those that are really shiny are very, very, very smooth, and that smoothness translates into this incredible shininess. So these unassuming birds have already invented nanostructured surfaces for their purpose. So that's really the nanostructural basis for the gloss of these Tinamou eggshells. This is also a Dan Hanley paper. Very, very fascinating. You might ask, how long have birds, or the precursors of the birds, the dinosaurs, been coloring their eggshells? Well, it turns out a very, very, very long time. There is a researcher, Jasmine Wieman, she's now at Yale. She looked at mass spectrometry evidence and Raman microscopy evidence for the presence of these pigments in petrified or otherwise preserved eggshells from hundreds of millions of years ago, and she found these signs for the presence of the pigments in a whole range of dinosaur eggs. And she was able then to put this into an evolutionary context. And so dinosaurs, or some of the dinosaurs, had green and blue eggs. And here these brown eggs. And again, this really shows, if you know what to look for, you can look for it with very sensitive techniques, and you may find it. To me, it's absolutely astonishing that these pigments still survive these millions and millions, these eons of time, that you can still find their remnants. Again, it shows the stability of the porphyrins and the oligoperolic pigments, the pigments of life. There is no coincidence that they were becoming so important in nature. It's really their absolute stability. Pattern. why are they patterned? Why are eggshells colored? Well, it's crypsis. It's hiding the eggshell from your predator who wants to eat them. And so here, the killdeer lays the eggs just on a gravelly beach, and they ought to be looking like the gravels they're lying among. And they're really beautifully colored and do this crypsis really well. Others lay their eggs on the ground into the green foliage, and they are perfectly adapted to the patterns of the foliage. Others are speckled and are in this bluish color, and you have to now think of, what light penetrates the dense canopy of the forest, right? Is it a white light or is it more a green light? And can I really see these these greenish eggs in green light very well? And the answer is they may be way more hidden under natural lighting than when you take a photo where you push all the leaves aside and want to take a photo. But then there are also these incredibly conspicuously colored eggshells that I've shown you before, right? Of the thrushes and the robin. And so why is that? Well, biology says, well, there may be other signaling going on. For example, if the hen lays very strongly colored eggs, it indicates it's a very healthy hen. So it's worth defending the nest. And so the males look at the bright eggs and say, oh, this is my offsprings, it's worth fighting for, they're healthy. And so the more colorful, the more vigorous the defense of the nest may be. That is one of the many signaling functions that could be also inherent to the color. And maybe the conspicuous color is offset by the advantages of having somebody really fight, defend, or protect the nest. There are structural effects that have been proposed in the literature, and I mentioned already the antimicrobial effects. Dan Hanley also went out and he looked at the eggshell color variation with latitude, and he finds that in the northern latitudes, the egg colors tend to be darker than in the equatorial latitudes or in Africa. So is there a thermoregulatory benefit of darker pigmentation? That's a question. These are the questions the biologists are asking, or the evolutionary biologists. And we as chemists, we just provide the techniques to quickly analyze and provide the tools for the analysis of the eggshells. There are beautiful patterns. There are, for example, these vermiculations on these eggshells that are, I find, fascinating. And what roles do they play? Well, there are some roles, and that is there are parasites. There are brood parasites. There are cuckoos that lay the eggs in the nest of a host, and then have the host do all the child rearing or the chicken rearing. And so there is no interest for the host to accommodate a cuckoo's egg. And so the host tries to mark their eggs so that they can distinguish it from the cuckoo's egg. And so there's this arms race between host and parasite. And, and then maybe if you develop the biology to put these lines on the egg, you can recognize your egg better from that of the host of the parasite. Here, for example, are 3 eggs of one type. This is of the warbler of the host, and there is here a large egg of the cowbird. And so we can see the difference. The bird doesn't, or maybe it does, and that's a question of biology. What differentiates one egg from another? How can birds recognize their own eggs? How can they recognize their egg in a colony if there are hundreds of the same eggs lying around? These are all the questions that are most likely related to the pattern. And it's just interesting to see the many roles of the pattern on the eggs. Some eggs are not colored at all. For example, the ostrich. Well, the ostrich puts the eggs in the wide open on the desert floor, so to speak. Shouldn't it be colored? Well, the ostrich is very capable of defending the eggs against any of the predators. And second is, they are laying the eggs under the African sun. And so the African sun really heats up the eggs, and you need as much thermal protection as possible. And a white egg reflects more of the heat than a brown egg would. And so biologists went and painted ostrich eggs brown and beige, and they died because they absorbed too much of the heat. And so here it's really good not to have a pigment on the eggshell. Here's an owl. The owl breeds in the center or in a hollow tree, and there is no need for crypsis if your entire nest is hidden away. And so there's no need for any patterning or coloring on the egg, and therefore a lot of the owls are having white eggs. So how does the color come onto the egg? How does an egg get formed? Well, the egg get formed, there is a follicle. It gets fertilized, it moves down the reproductive tract, the egg yolk starts growing, the various membranes grow around it. And then in the last hours, the shell gets put on. And when I say the last hours, this is relatively fast. And then the last putting on the shell takes perhaps a day. It totally depends on the bird. This is now for chicken here. So the eggshell gets formed in the last, say, day. And in the last hour or so, the coloring is put onto the eggshell. So, but how is it being put on? Well, for one, there's one interesting fact, and that is, in the beginning, the eggshell travels with a pointy end first. Then at some point, the eggshell turns around and then gets laid with the blunt end first. And so shortly before it gets laid, in the oviduct, there are some glands that put the coloring onto the eggshell. And here, for example, you see, you can almost see how it happened, right? As the eggshell traveled blunt end first, the glands on the side deposited a drop of ink, and it got streaked along the, as the egg passes down the oviduct. We can imagine this, and we can see this, and we can understand this. It's much harder to understand how does the bird get all this regular speckling on. And I have yet to find really good explanations how the coloring of eggshells works from a biological, from a mechanical point of view. How does that work? That you just deposit a very distinct ring on the eggs. Absolutely wonderful. So that means it's sort of a stop motion painting, right? You use the egg travels and stops and gets dots on it, and then it goes on. Here, these eggs with these very strong vermiculations, how does that go? If you try to follow one of these vermiculations, you try to unravel in which direction perhaps the egg moved in the uterus, you can't tell, because you would think that one gland and another gland, they would have similar patterns when the egg moves, but you don't find repeat patterns in here. So it's absolutely not quite clear how this is formed. Look at this. How many times did the egg need to move back and fro and right and left, and in how many different ways? Absolutely amazing, and not entirely clear how that is all being done, controlled, and really realized. Some of the eggs have the dye on a much throughout the eggshell rather than just on the cuticle layer on the outside of the eggshell, right? If you look at a hen's brown eggshell, you can take sandpaper and just paper off the brown shell, the brown from the shell. It's a very thin layer. It's the cuticle layer. But here there are some shells that are really through and through greenish. Here are 2 discarded shells or 3 discarded shells. Here is the biliverdin only on the outside, and here on these greenish eggs, it seems to be through and through. Notice the beautiful doily of the person that took the photo. I like that detail. Egg size. The egg size varies enormously. I mean, you have these egg sizes. Oops, you have the egg size of the hummingbirds, maybe the smallest hummingbirds, it's maybe 350 milligrams. An hen's egg is about 50 grams. An ostrich is about 1 kilogram. And here is the egg of the elephant bird, the Epiornis. Epiornis lived on Madagascar, is now extinct for quite some time. It was the largest bird that ever existed here, and it's about 10,000 or so fold larger than the smallest volume egg. That poses its own questions, right? Because the egg lives by the diffusion of gases inside and outside of the egg, and what works for a small volume may not work for a volume that is 10,000-fold the size of the small one. So really interesting questions as to the biology of the eggs. Eggshell shape. There are all kinds of shapes. There are papers in which the asymmetry and the ellipticity of the ovoid shape is being plotted against each other, and people try to figure out, what is the biological meaning of all of this? Well, some things are easy to understand, and one is eggshell size. If your chicken is already very developed in the precocial chicks here, well, the egg needs to be a little bit bigger because the development of the chicken is so much further ahead than in these species where the nestling or the bird hatches out of the egg in a very undeveloped state. So they can be much smaller. And then it really depends on the size and the shape of the pelvis, what eggs can be laid and what not. And of course, the shape of the pelvis, the shape of the bird, totally reflects what the bird does, how it lives, where it feeds, if it flies a lot, if it walks on the ground, if it migrates from South America to the end to the Arctic and back and forth each year, and so on and so forth. And so at the end of the day, the egg reflects an almost entire life story of the bird. And just by looking at the shape, the size, the patterning, and so on and so forth. There are also some very practical aspects. One of them is, if you're breeding on the edge of a cliff, you don't want your egg to fall down. So it needs to be very pointy so that when you knock it a little bit, that it doesn't roll off, but that it rolls just on a single spot. Whereas if you're breeding in a hollow, well, you can have very round eggs. They will never fall out of the nest, so they can be round. What is a pseudo-ovoid geometry? What is the geometry? How do you draw it? And I can refer you to some interesting YouTubes, how to draw egg shapes. It's not as simple as you think, and how many radii you have to combine to get to a really good egg shape. There are papers that are really taking this to an extreme on the geometry of the egg shape, and then classifying, the roundish ovoids versus the typical ovoids, and so on and so forth. And there's just a huge area of research out there trying to come to terms with the classification of eggs. Is the eggshell thickness the same all around? It turns out, no, it's not. Is the eggshell thickness dependent on egg length? Well, yes, somehow, and you can correlate it in one way or the other. You can look at deviations from this correlation in order to then figure out that environmental poisons were out there, right? We know DDT, for example, thinned the eggs of the birds, and they broke the eggs as they were sitting on them. And so that was the big problem of the apex predators of the bald eagles when DDT was still around. And so if you have a really good feel for the correlation between eggshell thickness and environmental pollutants, you can perhaps look much earlier at warning signs that pollutants are out there. So the embryonal development of the egg is complex. I'm not going to go into it. But what is clear is there needs to be an exchange of gases. It needs oxygen. The CO2 that gets generated needs to be exhaled. For that reason, there are these pores in the eggshells. There are gorgeously formed pores. There is an internal structure of the eggshells. Here on the inside, you see the membrane. And there are different polymorphs of calcium carbonate. The polymorph that is found in the eggshells is calcite. Aragonite, found in mollusk shells or so, is not formed. And then there is a very rare polymorph, vaterite, that is also formed on the outside of an eggshell of one particular species. And it was said that it is for collision protection because it's very soft. It's a layer that rubs off very easily, as you can see on the progression from left to right. The eggshell structure is different. There are different grain structures going from the inside to the outside. And it is a fine balance between stability and, at the end, the chicken has to be able to break the eggshell and hatch. How coarse the crystalline structure of the calcite is controlled by a protein called osteopontin that's also found in bone, controlling also the crystallization of calcite in our bones. And so if you have no osteopontin present, you get very fine, smooth structures with very large crystals. And the more osteopontin is present, the finer the crystals. There's also beautiful chemistry. Calcium carbonate plus bicarbonate is in equilibrium with the calcium bicarbonate. The bicarbonate dissolves, and it can dissolve slowly the eggshell, and that can be taken up by the chick. And so a large portion of the calcium in the bones of the chicken developing in the egg comes from the eggshell. And about 10% of the eggshell mass came from the bone structure of the mother, of the hen. And so it's a direct transfer of calcium from the bones of the hen to the chicken, to the chick. Sorry. And so as the chicken develops and exhales CO2, it softens the calcium carbonate and dissolves it slightly, and then eventually the chicken can come out. It can break out, and we all have seen probably these pictures of the little egg tooth to break through the shell. I think it's much less for the breaking through the shell than breaking through the inner membrane. The shell, after the eggs were incubated for a long time, get very, very brittle, but the membrane is still very tight and tough. And so there's this little egg tooth that cuts open the membrane. And you can see this, here's the egg tooth on a turtle. And the turtle has a soft egg. There's no calcite layer, but it has all membrane. And so it also needs a little tooth. Last not least, we like to eat eggs. And there's one color that I could not leave out when talking about eggshells and eggs, and that is the yolk color. The yolk color is, unlike the eggshell color itself, not genetically determined, but it is to a very large degree nutritional. So it's carotenoids that color the egg yolk. And if you feed your chickens paprika extract in marigold, or you can also generate these orange egg yolks. So I've gone around and around. I have collected over the years the books on eggshells and have been reading them, but there are two recent books that I can recommend. One is by Mark Harbour. It's the Book of Eggs. It's just a big collection of beautiful, beautiful eggshell pictures and a lot of information about them. And then there is another book here, The Most Perfect Thing, by Tim Burkhead. It's more a narrative about birds' eggs and the history of how they were collected, and it makes a really good read before going to bed. That is it for me. I'm already a couple of minutes over, and I'm more than happy to answer all your questions. Thank you. I will stop sharing because then I will see you again.
Thank you very much, Dr. Brückner. We have a couple questions here available for you. One is, if many hens make colorless eggs, then why do some other hens have the need to make color eggs? In other words, if those hens that make colored eggs with those polyphyrins, do they actually make another use of those polyphyrins? If not, then why color the eggs if they don't have to? And for us to listen to a pretty interesting talk about eggshells, and that is from Olivier Niquez out of the New Haven ACS local section.
That's a good question. And the question is, what is the natural color, right? And when do we get white eggs? When do we get brown eggs? And my suspicion, and I'm not entirely sure about this, my suspicion is the brown eggs are the natural color, and the lot of breeding that we have done generated the white colors. Because there was no further, there's no pressure for it to be colored, and they could get away with the white eggshells. But I'm not 100% sure. But it's, the hens that we know today, that we breed for eggs and meat, are really not very natural anymore. And so a lot of things may be different than found in natural environments or natural hen environments.
Thank you. We have a couple other questions. The Rye High Chem Club has a question: can eggshell colors change over time?
They do, and some quite a bit. The Tinamous that I showed you change their color very quickly. These pigments that we discovered are very light sensitive, and so they change their color very quickly. And it is related to how the Tinamous breed. So several hens of the Tinamous lay their eggs in the same nest. And so the hen needs to time very well how old the eggs are that are already in the nest. Because if they are very old, well, then there's no point in laying new ones in, because then the first ones hatch and they'll probably abandon the older eggs. So you want to have a cluster of same aged eggs, and maybe the color is really an indicator how old the egg is. You can also take a brown eggshell, leave it for a month or so on the windowsill, and then look at it again, and it's much paler. It really bleaches.
We have a couple other questions. Are polyphyrins the pigment of choice just by convenience? What is the matrix for pigments that is applied to eggs? Are there proteins along with the pigments?
Yes, it's a protein shell. It's the cuticle. I don't know much about the protein itself, in which the porphyrins are embedded, or the biliverdins, but there must be something important about them, because the biliverdin colored eggs have all different kinds of colors, from dark green to this light blue. And it's not just a matter of concentration, it's also a matter of, I think, conformation, and the protein will adjust the conformation and therefore adjust the color. So I would love to learn more about that, but we haven't. So it is, yes, there is a protein matrix involved, but I know nothing more than that.
Okay, we have one other question regarding the photosensitization experiments that were displayed in the 1930s. How can scientists study and tune the phototoxicity of these molecules?
They are already very phototoxic, and so the tuning of the phototoxicity is usually not what we are aiming at, but what we are aiming at is the tuning of expressing the phototoxicity in exactly the cells that you want them to be expressed. If you inject a drug and you just treat a tumor in one side of you, but the entire body becomes photosensitized, that's not so good. You want something that's sort of the silver bullet, that it goes just to your tumor, then you shine light only on the tumor, that only kills the tumor, and then the patient can walk out in the sunshine and not get hurt. And so it's the selectivity of the phototoxicity that is a much bigger problem, and that's what people work on tuning, than the photosensitivity itself, or the sensitization ability.
Great, and we have one other question. What was the evolutionary advantage of the shiny eggs compared to the same species also making matte ones?
I really don't know. I don't know. The Tinamous are a fascinating bit. But I really don't know if anybody has figured out the evolutionary context there, or if they are shinier and they are more vigorously protected or so. I don't know.
Yep.
And as far as I know, the papers on the shininess are silent on this as well. So, but that's one of the big difficulties of the biology, right? Putting things into a biological context, in a evolutionary context. It's so much easier just to point at the pigment, but then how to put the pigment into context is a big issue.
Oh, and one more question that just came in. Any details on calcium flux in a hen? Is it a lot?
Calcium flux, yeah. So about 10% of the eggshell is coming from the bones of the hen. And then, and I forgot the exact numbers, a good portion of the bone structures in the chick come from the eggshell. And I guess most of the data, 99% of the data in the literature is all chicken, right? Chicken breeding is a big thing. And so I don't know if that's true for every species, but that's about it. So there is a significant flux of calcium from the hen to the chick through the eggshell.
Thank you very much, Dr. Brückner. I'd like to give everyone a virtual round of applause for coming out and speaking with us this evening. Very much appreciate your time. And if there are any additional questions, you can always send them either to myself or Tom Burkholder, who is also the webmaster for our meeting, and we can forward them on to Dr. Brückner as well. And one announcement I would like to make is that coming on November 11th, we do have a talk by a Mr. Stephen Geppert, who is a retired fisheries biologist from the state of Connecticut, DEP, who will be talking about the imprinting homing mechanism of anadromous fish. And thank you everyone for coming tonight. It was much appreciate your time. And again, thank you, Dr. Brückner.
Oh, thanks again for the invitation. It was a pleasure.
It was a pleasure having you. Thank you so much.
All right. Thank you. Bye-bye.
Okay. Bye-bye now.